Dear student, today we will discuss C4 pathway, which is also part of dark reaction. C4 pathway is also known as Hatch-Slack pathway, as it was discovered by Marshall Davidson Hatch and Charles Slack. The first stable intermediate formed during the pathway is 4-carbon compound, so it is known as C4 pathway. The plant in which the C4 pathway is operated are called C4 plant. Now, where all or which all plant are known as C4 plant? Let's talk about their adaptive features. These plants grow in high light intensity, such as tropical plant, sugarcane, grasses, maize, etc. So they can tolerate high temperature and they have a tendency to fix extra carbon dioxide. Therefore, they can even fix the carbon dioxide at low concentration of carbon dioxide. Usually, if the carbon dioxide is lower and oxygen concentration is higher, the photorespiration is favored. So, C4 plant do not favor photorespiration under any circumstances. Now, they have a special leaf anatomy called Cringe anatomy, and in this, the mesophyll cells are clustered around the bundle sheath cells in a ring-like fashion. The number of chloroplasts in the bundle sheath cell is more than that of the mesophyll cell. So you can just have a look, upper and lower epidermis, and then vascular bundles are there and they are surrounded by these bundle sheath cells. So they are special cells which have more chloroplast, and you can see the mesophyll cells are also surrounding. Otherwise they used to be present as a palisade cell, but here they are surrounding the bundle sheath cell. So this is a special anatomy of C4 plant, and this is known as Cringe anatomy. Now we will see what happens in. So it occurs. Okay. So it occurs in mesophyll cells and bundle sheath cell. Two cells are involved. So if you look at the mesophyll cells over here, the acceptor of carbon dioxide is phosphoenol pyruvate. And look at the structure, which is a 3-carbon compound, and this phosphoenol pyruvate in presence of enzyme PEP carboxylase is converted into oxaloacetate. Then oxaloacetate is converted into malate. So again here the reducing power is used, and then malate is translocated to the bundle sheath cell. Now in the bundle sheath cell, the malate is converted into pyruvate. Again reducing power is used up here, and the pyruvate is again transported or translocated to back to the mesophyll cell. Now here the pyruvate is converted again back to PEP, that is phosphoenol pyruvate, by using ATP. So this cycle is known as C4 pathway. Now here you can see malate, which is the 4-carbon compound, pyruvate, which is the 3-carbon compound. Okay, so one CO2 is released over here, and this CO2 enters the Calvin cycle for the fixation of carbon. So extra carbon dioxide is fixed up over here. Now if you look at the difference between the C3 and C4 plant, then in the C3 plant, mesophyll are the only cell that are involved in carbon fixation, whereas over here mesophyll and bundle sheath cells are involved. In C3, the CO2 is fixed once, it is fixing up to 2 times. Over here there is no Cringe anatomy, whereas C4 plants are associated with Cringe anatomy, and CO2 is the acceptor for Ribisco. Here CO2 is accepted by PEP carboxylase, and second acceptor is Ribisco, because Calvin cycle is also going on here. Okay, so this is all about C4 plants. Thank you.